Hey all, so today we're going to be looking at SL and HL content for A3.1, which is evidence of evolution, looking at pages 97 to 115. And we're going to be figuring out what is a species and then what patterns are seen in the diversity of genomes within and between species. So not surprisingly, our theme here is unity and diversity. So species, variation occurs in all individuals, and this is a foundation of evolution. Natural selection needs variation. And species have similar characteristics. Carl Linnaeus created the classification strategy that we still use today, the kingdom, phylum, class, order, family, genus, species, binomial system. And he did that using morphological characteristics, so physical characteristics, because DNA mapping was obviously not a thing then. And so when we write it, we write the genus name first, capitalized, and then the species name. And that would be in italics if you were writing, typing it. But if you are writing it, then you underline it, because obviously, how do you write in italics? And then a genus is a grouping for species. And so a biological definition of a species is a species being a group of organisms that can interbreed and have fertile offspring. This usually holds true, but due to geographic isolation might not be true. So obviously, tigers and lions both belong in the grouping Felidae, and that's their order, but their genus and species are different. And lions and tigers can reproduce if they're put together. They can produce a liger if the lion is male, or a tigon if the tiger is male. But almost uniformly, male ligers and tigons are sterile, and only occasionally can the females reproduce. As well, ligers and tigons have health issues and often die before their parents would normally. So speciation is often due to geographic separation, and a population of a species begins to gradually change due to the fact that they can't or don't interbreed with the original population. This process is called speciation. And so this is what Darwin saw with his finches on the Galapagos Islands, which is what promoted him to write The Origin of Species, which is the book outlining the process of evolution through natural selection. And so there was some OG finch in Ecuador, the mainland finch, and it blew off course in a storm and ended up on the Galapagos Islands, some of them. And so over time, these finches changed their feather colors or their beak colors to be better suited for the environment that they were living in. And obviously do not interbreed because they're on different islands. However, if the two populations hadn't yet changed very much physically or genetically, and were still quite similar, they would be still part of the same species and could interbreed if they were put back together. So at what point do scientists decide that these are two species that now we can classify them separately is difficult, because obviously they sometimes are not interbreeding because of that geographical isolation. So they're, unless we were to move them, then they wouldn't be interbreeding in anyway. So chromosomes, each species have the same number of chromosomes. So like all humans have 46 chromosomes, and 23 of those chromosomes come from their mom, and 23 of those chromosomes come from their dad. And changes can occur. So like you can have fusion or splitting or doubling of a chromosome can happen, but it's really rare. And usually a species will stay the same for millions of years. Haploid cells are sex cells, and they only have one set of chromosomes, and that's so that their one set can merge with another set to make a full set. So we say that those are 1N, or haploid. And body cells have two sets of chromosomes, meaning they're diploid, and they have half their chromosomes from mom and half their chromosomes from dad. And so there's 23 chromosomes in a human body. Chromosome 1 has 2 copies, chromosome 2 has 2 copies, all the way up to chromosome 23. And the number of chromosomes isn't really important as long as the species numbers have the same number. So, for example, horses and donkeys can interbreed, and they do not have the same number of chromosomes. Horses have 64 chromosomes and donkeys have 62. And so they can make mules, which have 63 chromosomes, but then mules are often sterile because they have an odd number of chromosomes, and so they can't match up properly with other, they can't do the meiosis process properly. And so that can make it really hard for mules to reproduce. But with chromosomes, you could have 2 big chromosomes or 8 small chromosomes. It doesn't really matter as long as you have enough genes to code for the characteristics that you need in your body. It doesn't mean some organism is more evolved because it has more chromosomes. Karyotypes are diagrams of chromosomes during the mitosis process. So when cells are replicating, so they've condensed their DNA, and then you can see the different chromosomes appearing. Genomes are all the genetic information in an organism or group of organisms. So this is all the bases in their DNA. And genes are lengths of DNA which can code for specific proteins. They're always in the same place in a species. So for example, if we were talking about horses, then horses have 1 chromosome from mom, 1 chromosome from dad. And let's say the top of my finger here is the gene that codes for fur color. And so they can be swapped. Those 2 genes can switch places on the chromosomes. That's called crossing over, and that increases genetic variability and diversity. But you can't switch like this part down here with that part up there because those are 2 different genes that code for different characteristics, as opposed to being 2 different alleles, which are alternate forms of a gene. So for example, A, B, and O blood are all different types of the same gene that cause blood type. But A, B, and O are just caused by a small change in the base sequences that result in a different allele. So single base changes are called single nucleotide polymorphisms. There's a lot of them, about 100 million of them in the human genome, approximately 1% of the bases. So any given individual might have 4,000 to 5,000 single nucleotide polymorphisms. And this causes diversity in human populations and leads to different alleles. Genome modifications. So genome size is measured in base pairs. Big genomes don't necessarily contain more functional genes than small genomes. Non-functional DNA are sections that don't code for proteins. And transposons are segments of junk DNA that are called jumping genes. And in prokaryotes, they can be cut out and moved around. And this ability is mostly lost in humans. But initially, when scientists were first studying DNA, they didn't understand what certain segments of the DNA did because it wasn't coding for proteins. And so they called a bunch of different stuff junk DNA. But we now know it has a bunch of different tasks in the cell and in the body. Genes have critical functioning. So they will make proteins and genes that do have critical functions, for example, cytochrome C is critical for respiration. And these genes tend not to change very much over very long periods of time. But other genes might change as populations become isolated due to natural selection. So that could be something like eye color is changing or hair color is changing. Those are not critical for life functions. So all human base sequences was mapped in about 2003, and there were 23,000 genes, which was about 3 million bases. This process has become way cheaper now, so about 10,000 times cheaper than it was in 2000. And so obviously many more species are being sequenced, and the goal is to investigate the evolutionary origins of different species. Mapping all human individual genomes allows us to know migration patterns and info about genetic diseases, and that leads to potentially personalizing medicine. So when the genome was being sequenced, what they would do is replicate the DNA using these fluorescent base tags. And then they would load this into a gel electrophoresis tray, and a computer would analyze the fluorescent tags so they would know where do the C's occur, where do the G's occur. Most of the genome doesn't actually make proteins. So it's satellite DNA or non-coding DNA. Non-coding DNA can make telomeres, which is the end caps on on chromosomes, centromeres, which is a connection point to other strands of DNA. It can be short tandem repeats, which are segments of the same bases that repeat over and over again. And those are used to identify individuals because your short tandem repeats are unique to you and your family. But also segments of DNA can be promoters or silencers, and also can be used to make ribosomal RNA or transfer RNA. So for SL, to summarize, species can interbreed and have fertile offspring, and they usually have similar characteristics. All species have the same chromosome number. Genomes have segments that make proteins as well as non-coding segments, i.e. transposons. And genes can mutate single bases. So there is some variation in any population. These are the SNPs, and then but that usually makes little difference to functionality. Genes that are critical to life functions don't tend to change. Alleles are variations of a gene, like ABO blood. Mapping of the human genome occurred in 2003, but there's a lot more sequencing of DNA happening now to look at evolutionary pathways. And I mentioned previously a karyogram. This is a karyogram of a human individual. And so you can see that there are two of each of the different chromosomes. All right, if you're SL, you can stop watching at this point. If you're HL, we will continue on. So asexual reproduction, some organisms are not considered species under the biological species concept because they no longer sexually reproduce. They're clones of one another, so they're asexually reproducing, and therefore their DNA is not mixing to achieve a middle unified group. So an example of this would be blackberries or dandelions. I had no idea before I read your textbook that these were asexual reproducing organisms. Usually species branch off from each other, becoming separate species. So we can see this here as a cladogram showing common ancestry here, and then all the branches occurring as variation is happening. But if horizontal gene transfer is possible, such as with bacteria from one species to another, it's hard to know if the biological species concept works for prokaryotes because a lot of the genetic material is being swapped around. And so are they sexually reproducing? No. Are they transferring genetics? Yes. So that it enters into a bit of a gray zone. This sometimes happens in eukaryotes, but it's very rare. Certain species having a set number of chromosomes is reduction in diversity due to sexual reproduction. So they just have to have the same number of chromosomes to reproduce effectively. And so, for example, talking about mules again, mules are genetically less likely to be able to reproduce because they have an odd number of chromosomes. So horses can reproduce with horses because they have the same number of chromosomes. Gametes have half the number of chromosomes, so they can join together to make a full set of chromosomes to make offspring. And making gametes happens in meiosis. One chromosome's from your mom, one's from your dad, and those are called homologous chromosomes. And if two organisms with different chromosome numbers had offsprings, they would likely have problems making fertile gametes because you would end up with a gamete that has 3 chromosomes as opposed to just 2, so that you have a duplicate of 1. And this is actually what happens for humans who have Down syndrome. Trisomy 21 is where the 21st chromosome doesn't separate properly in meiosis. And so in the egg cell, typically, you end up with 2 copies of it. But the thing is, with chromosome 21, it's quite a small chromosome. And so it's still possible to have a functional human who has been born and has a reasonable lifespan as well and relatively healthy, despite having an extra chromosome. So dichotomous keys are used to ID species within a group. And so dichotomy means division into 2. One description should clearly match the species and the other should be clearly wrong. And this can be used to ID species. So there should be clearly visible features that are reliable. And so if we were trying to identify vertebrates, we would say, does it have fur or no fur? If it has fur, it's a mammal. If it has no fur, does it have feathers or no feathers? If it has feathers, it's a bird. So you can see here, these different characteristics allow for identification of these different classes of organisms. And here we can see a similar dichotomous key using invertebrate phyla, asymmetric versus symmetric, bilateral versus radial symmetry. So certain segments of DNA can be used as a barcode to identify species. And usually, interestingly, these segments of DNA can also be found in the water in the ecosystem that these species are present. And so it allows scientists to detect which species are in the environment without actually having to go out and count species in the environment. So prokaryotes and asexual reproducers don't follow the biological species concept. Partnered homologous chromosomes allowed fertile offspring to be produced that can make haploid gametes through meiosis. Dichotomous keys can be used to ID species in an ecosystem or to differentiate between categories of organisms like phylum. And DNA barcoding can be used to determine the species in an organism as well. And so here, these are our blackberries and dandelions that are reproducing asexually. Okay, that's it for this video. Thank you very much for watching.